So the third topic I want to talk about in just the last 20 minutes or so are these tightly coupled computations. These are the hardest computations to try to parallelize. The standard example is you have some sort of graph or you have some sort of network, and in every phase of your computation, every node in your network is going to try to talk to every other node in your network. So here I have, blue turning to green, turning to red, as these different signals are being processed. Traditionally, the way that one would try to tackle this sort of graph problem would be to use MPI to get the different computers to talk to each other. But it's also possible to use MapReduce to do the same sort of analysis. In fact, this is exactly how PageRank is computed in a MapReduce framework where different nodes in the web graph are able to talk to each other. So again, going back to this dance metaphor, if we started with this very simple dance that was embarrassingly parallel, then we had this more complicated dance that was our loosely coupled scenario. Now with these tightly coupled algorithms, we have to choreograph a very, very sophisticated, highly stylized dance where your different computers, your different workers are able to synchronize in just the right way. Now before I tell you about the dance that we're going to do in a parallel setting, I'm going to give you a little bit of analogy to try to make it clearer, why and how this dance may develop. So the analogy is going to be an analogy for that problem of genome assembly, but instead of reconstructing genomes, we're going to imagine this problem of trying to reconstruct a book. So imagine you're Charles Dickens and you just finished writing A Tale of Two Cities, and that begins, it was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness, and so forth. So Charles Dickens just printed up his book on these very, very long spools, and as he walks away from the copier, he trips, and then the spools of text get chopped up into short fragments. So the metaphor is, we have our long genome, and then through the sequencing process, we're just going to be able to read little fragments of that genome. So here we have many fragments, each with 5 words per fragment. So then the question is, how could you reconstruct the text from all these different fragments? So if we started with 5 copies of the book, each book has about 140,000 words. That means we have about 140,000 fragments. All those fragments get mixed together in the mixing bowl. And then just one thing I want to point out is some of these fragments are going to be identical. So of times it was the occurs in different places. It was the age of occurs in different places. So even though it's getting chopped up, these different fragments can have the same words on them, even if they occur from different places in the original text. In the case of a genome assembly, this sort of repeated sequence occurs all the time. So then the question is, how can you reconstruct this? So here I've just taken those fragments and laid them out alphabetically. The first thing that you might try might be like a greedy reconstruction. So maybe Dickens remembers that, the very first word of the book starts with a capital letter. So we're going to take a fragment like this, and then we're going to try to find the next best fragment. So in this case, the next best fragment was the best of times, and it's the best in the sense that it has the best overlap, where an overlap here is the end of the first fragment perfectly matches the beginning of the next fragment. We could have imagined picking a fragment that just overlapped by a single word, but generally we want the longest overlap as possible because we want these pieces to be as connected as possible. So this is great. So we started with 5-word fragments, but now we have 2 of those, and that spells out 6 words of the original text. We can add the next fragment. Now we spell out another word, then another word, then another word. So now we've spelled out it was the best of times, it was the. Notice at this point there are multiple, in this case 2 fragments that overlap equally well. That's not a problem in itself, just something to take notice of. But then on the very next attempt to apply this greedy algorithm, we have a conflict, where we have 2 fragments that overlap equally well, but it's really not clear how the text should be reconstructed. It's not clear if it was the best of times, it was the worst, or it was the best of times, it was the age. There's just not enough information from the sequence fragments to be able to reconstruct the genome as a whole. So rather than trying to do some sort of greedy approach, we can model this sequence reconstruction problem as a graph problem. So the graph that we're going to construct is called the Bruijn graph, and it does something somewhat unintuitive. So we're going to take fragments that start, in this case, 5 words long, and then we're going to decompose it into even shorter subfragments with directed edges. So we're going to take this 5-word fragment, decompose it into these 2 4-word fragments with a directed edge in between this. So the interesting thing, and we're just going to do this for all the fragments that we have. So the interesting thing is doing this really simple operation about taking fragments, decomposing them, is going to reveal the global sequence structure, and it's going to capture all the overlaps between the sequences. So on this next page, I'm just going to do this simple algorithm, this simple operation, where we're going to take those original fragments, we're going to chop them up into subfragments, and then lay them out just on a piece of paper using, some sort of graph layout algorithm. So hopefully you can see the power of this Bruijn graph framework is that you have what was used to be all those isolated fragments where it wasn't clear who was connected to who. Now we have it laid out into this nice picture where we can really see the global structure of the fragments. Now since we're trying to reconstruct the book, or in the case of genome assembly, we're trying to reconstruct that genome, a natural thing to do next is to take all of those individual fragments and parts of the graph where it's one fragment followed by another fragment followed by another fragment, and then merge those all together into larger pieces. So we're going to do that, and that'll simplify the graph like this. So now we have this nice long fragment, it was the best of times it. We have other nice segments where we've been able to reconstruct the original book in as clear a way as possible. If we had all the fragments from the entire book laid out, we would be able to find a tour through the graph that exactly matched the original text that was there. It just becomes a question of finding what's the right tour through the graph. Now we might be tempted to do operations like, and notice that there's a little cycle here. We may be tempted to do things like try to collapse this cycle into the graph so it's spelled out. It was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness. We might be tempted to do that, and that's because in English it would be very awkward to write, it was the best of times, it was the worst of times, it was the worst of times, it was the worst of times, it was the age of wisdom, it was the age of wisdom, it was the age of wisdom, it was the age of foolishness. It'd be awkward in English to have those little repeats right next to each other, but in real genomes that sort of motif occurs all the time. So we have to be very, very careful to carefully model the number of copies that need to be present, and then have a correct algorithm that can place the right number of copies.